Hello and welcome to your Chapter 7 video lecture, which is going to cover the nervous system, specifically discussing the structure and control of movement. And here is our outline for Chapter 7. We'll talk about the general nervous system functions, organization of the nervous system, sensory information and reflexes, muscle chemoreceptors, somatic motor function and motor neurons, vestibular apparatus and equilibrium, motor control functions of the brain, motor functions of the spinal cord, control of the motor functions, the autonomic nervous system, our motor unit aging and the preventative influence of exercise training, and exercise enhances brain health. So again, starting with our general nervous system functions, we can divide out our nervous system into a central nervous system and a peripheral nervous system. Focusing on our central nervous system right now, it's going to help control the internal environment of our body. It really helps to coordinate along with the endocrine system, which is going to involve our hormones and allows us to have voluntary control of our movement, specifically controlling our skeletal muscle. And we see that this is integrated within our spinal cord in order for us to have reflexes. So notice that our central nervous system is made up of the brain and the spinal cord. So the spinal cord is kind of a highway where we can communicate with the brain, either giving input in or sending information or tasks out. Within this system, we'll also have assimilation of experiences necessary for memory and learning, meaning that we have a process to fully take in and understand information or our ideas. So as I mentioned, our central nervous system is made up of our brain and spinal cord. But what about our peripheral nervous system? That's essentially going to be everything else outside of the central nervous system. So we'll have some peripheral spinal nerves. We will have our cranial nerves involved within the system. We do divide it out into what we call a afferent or sensory division, and a efferent, and I'll just be really pronouncing those E's and A's so it's clear. So efferent, even though we would say efferent, and this is also called our motor division. So the way I remember that sensory means afferent and motor means efferent is by using the word same. So SA for sensory and afferent, and ME for motor and efferent. So when we're talking about our sensory division or afferent division, this means that we have afferent fibers that are transmitting impulses from receptors into the central nervous system. So the receptors could be associated with somatic sensory neurons, visceral sensory neurons, or special sensory neurons. So somatic, you want to think information that is really coming from your skin, your muscles, your bones, and your joints. And then we have our visceral sensory. So anytime we see that word visceral, you want to think organs. So that's going to carry signals mainly from the organs of our thoracic and abdominal cavities, something like the stomach or our heart. And then we have our special sensory pathway, which is going to bring in information from areas of our special senses. So for instance, our ear, or from our tongue for taste, or nasal area for sense of smell. Our motor division or efferent division is going to have fibers that transmit impulses from the central nervous system and move over to our effector organs. So this would include our somatic motor and autonomic motor areas. Somatic motor is really going to consist of our skeletal muscles, whereas autonomic motor, that's going to deal with our heart, so cardiac muscle, our smooth muscle, so areas such as the digestive organs that have smooth muscles within them to contract and move food along, or blood vessels that have smooth muscle in there to help move blood along. And the last effector organ would be our endocrine glands or exocrine glands as well. So allowing substances to move through a duct and into other areas, like a sweat gland, for instance, would be an exocrine gland. Allowing that sweat to move through the duct and have your sweat stay on your skin and evaporate to cool the body down. Or, like I said, an endocrine gland. So having your thyroid gland secreting thyroid hormones, that is a part of the autonomic nervous system. So note that we do have this under a specific type of nervous system that controls that. So within our autonomic nervous system, we would have our sympathetic nervous system, parasympathetic nervous system, and our enteric nervous system. We'll elaborate on sympathetic and parasympathetic soon, but our enteric, let me just mention now, is kind of a brain of its own for our gut. It's going to help control our process of digestion. And here's another figure in your book showing you first the structural organization of our central nervous system, the brain and spinal cord, and as we said, everything else belongs to our peripheral nervous system. So we're going to have our nerves, and then we also have structures known as ganglia. Ganglia are going to be areas where we have collection of cell bodies. So if I were to have this area of a bunch of cell bodies, this is what our neuron essentially looks like. So we would have all of the main area here, known as our cell body, in one place. And typically on the body, this looks like a little bump like that. So that's called the ganglia when it's found in the central nervous system. Now, when we find an area like that in our brain, this is a really terrible looking brain over here. If we find an area where we have all of these cell bodies in the brain, then that would actually be called a nucleus. So I know we use the term nucleus for our cells, but the term nucleus is also used to refer to a collection of cell bodies within the central nervous system, within the brain. All right, now let's go back to talk about the functional organization. As we said, we can take that peripheral nervous system and set it up as our sensory nervous system and motor nervous system, or divisions. And so our sensory nervous system, we said, is going to detect stimuli and transmit information from a receptor to the central nervous system. And we're essentially doing that in the somatic nervous system through our skin, through the muscles, your joints. And I know here it says the eyes, ears, and skin. So they have also lumped in the special sensory in this category. So that's why we've got everything listed here. With our visceral sensory, again, we're taking in information from our organs. So we're taking in information from our blood vessels and other internal organs such as the heart, our lungs, our intestines, and urinary bladder, for example. Motor division, once again, we are taking information from our brain, moving it down our spinal cord, and then it's going to move on out. So somatic motor division is going to send information out to our skeletal muscles, whereas our autonomic motor division is going to send information out to our smooth muscle structures, our cardiac muscle structures, such as the heart, and our glands. So those could be endocrine, involving hormones, or exocrine, involving ducts that would secrete substances. Now let's revisit some information that you probably discussed within an anatomy and physiology course, but just as a refresher, let's look at the structure of a neuron. So here we have our cell body, and our cell body is going to contain the nucleus and majority of the other organelles within our cell here. So remember, even though we talk about cells as being these circles and having the nucleus and all these organelles, the neuron is still a cell, right? We're going to have our mitochondria in here, a golgi apparatus, and we'll be having vesicles moving around. And so we don't want to lose that, but we also know that a neuron has special features such as dendrites. So these are going to be cytoplasmic extensions that stick out from the cell body that will be able to conduct impulses toward the cell body and then direct them into this axon hillock. It's kind of where all the information gets gathered. And then we can carry electrical impulses away from the cell body through the axon. So that's this inner layer in here is the axon, but we may have the axon covered by what I like to think of as almost like hot dog buns called myelin sheaths. And these myelin sheaths within the peripheral nervous system are covered by a Schwann cell, but in the central nervous system, it is actually put there by another cell known as an oligodendrocyte. But in both situations, we are going to form this discontinuous row of myelin sheaths along the length of the axon, and this is going to help to move that impulse through much faster than if it was unmyelinated, if these myelin sheaths did not exist. I always compare this to if I made you walk and have to touch every single part of the floor to get to the other side of the room versus if I allowed you to run, and in that way we are kind of jumping in the air, right, and skipping portions of the floor, and so we are able to reach the other side of the room much faster. And then we reach the end of the neuron, and here at the synaptic terminals, we are going to have synapses take place. So essentially, and I'm going to switch to another figure to explain this, essentially we have moved that impulse through the first neuron, and now we are going to be at the synaptic terminal and allow that information to move across this space, which we call the synaptic cleft, and over to a dendrite of a second neuron, which we are going to call a postsynaptic neuron because it's post after that synapse has moved through, and we can move the impulse through the second neuron. Now let's take a little break to talk about a clinical application here and discuss multiple sclerosis or MS. MS is a neurological disease that destroys those myelin sheaths we just described on our axons, and it does occur due to an immune attack on the myelin. So in the peripheral nervous system, they would be on these Schwann cells that create the myelin sheaths. We don't know the exact cause, but the theory is that there are defects in several genes to cause this to take place. So if someone has MS, there is a progressive loss of the nervous system function. And that's because we're not having the impulses moving through these neurons as quickly as they should. And that can lead to fatigue, muscle weakness, poor motor control, loss of balance, and can lead to mental depression. So there are many other mechanisms at play to cause all of these symptoms to arise, but we will keep it simple and just focus on that impulse not moving through the axon. Now, what we have found through research is that exercise training will improve both the functional capacity and the quality of life for these patients, and is serving as the most important non-pharmacologic disease treatment for MS. So those patients that are exercising regularly were shown to have increased strength, endurance, and fewer symptoms. And the mild, moderate forms of MS benefit from generalized exercise prescription. So that would be with 2 to 3 days of exercise per week for about 10 to 30 minutes a day with moderate intensity, and doing 2 to 3 days per week of resistance training. That would include 1 to 3 sets and 8 to 15 RM. This will also help to maintain and improve flexibility and emphasize fine motor control. And then there are patients with the severe forms of MS. They may need supervised training to prevent injuries since they may have more poor balance or loss of their motor control. Now let's review electrical activity within neurons. Neurons are always described as being excitable. So we can describe being excitable in two different ways. First is their irritability, their ability to respond to a stimulus to trigger a nerve impulse to move through. So here's our example of touching the flame within a candle. Obviously that's going to burn and cause pain, so we can trigger that neuron here to send information into our spinal cord to trigger a reflex going to our muscles to pull our finger away. And then we have conductivity, which is going to be the transmission of that impulse along an axon. So as we were talking about the structure of a neuron, that impulse moving through, and this would really have to do with more information regarding the internal environment of the neuron. So let's discuss that next. When our neuron is at rest, it is going to have a negative charge inside of its cell, and that can range from -5 to -100 millivolts across all cell types. So if we're looking at liver cells or skin cells or our enterocytes, which are our intestinal cells. But when we look at neurons specifically, we see the resting membrane potential can range from -40 to -75 millivolts. So again, we're talking about inside that neuron. That charge is determined on permeability of that plasma membrane. So remember, that serves as the boundary of the neuron and determines what we're letting in and out. So are we letting sodium move in? Are we letting potassium move out? So the difference in the ion concentrations across this membrane play an important role. We are going to see that we have more, and I'm going to switch to a different figure for this. We are going to see that we have more potassium inside the cell, and as far as outside of the cell, within our extracellular fluid concentrations, we will have more sodium, more chloride, and more calcium in those areas. So what helps to maintain that is something called a sodium-potassium pump. If we look along our plasma membrane, we could see that pump right over here. And we don't have it pictured here, but imagine another little channel over here, which is going to be called a leakage channel. This leakage channel, just as the name sounds, tends to allow potassium to diffuse out of the cell. So we would start to get potassium building up here in the extracellular matrix. But the pump then takes that potassium and we'll pump it back into the cell, so we can increase the potassium concentration inside of the cell. And in exchange, we will have 3 sodium ions move out of the cell. So sodium would increase out here, helping to formulate what we see at rest. More sodium outside of the cell and more potassium inside of the cell. Now there are channels that help to regulate this ion passage across our plasma membrane. So this is depicting for us an open channel where we can have these ions flow to the extracellular space, and then we can also close that channel so that substances cannot move through. Sometimes these channels are controlled due to their voltage, and other times they are going to have little areas where a ligand or a chemical substance will have to bind in order to cause it to open, and other times it's going to be due to a mechanical change within the cell. Maybe there's a stretching of the cell that would take place. So we've talked about a neuron at rest. How are we going to transmit information through the neuron? We do this through something called action potentials, where a stimulus of a sufficient strength will depolarize the neuron. Now I want to describe that term depolarize for you. When we think about the word polar, and to think of the definition of that, let's talk about a water molecule. So water, if you remember from a chemistry course or maybe like an introduction course, is going to be made up of an oxygen atom and our 2 hydrogen atoms, right? I won't write the little H's in here, maybe just one. So that oxygen is going to have electronegativity. If you think back to, I think it was chapter 1 or 3, we talked about the valence electrons, or the electrons found in the last shell of atoms. And oxygen wants 2 electrons to be stable and happy and fulfill that octet rule. So that is going to cause oxygen to borrow or share the hydrogen electrons, but it wants to keep those electrons a lot closer to itself. It's kind of greedy, and we know that electrons have a negative charge to them. So we will have more negative charge toward this oxygen, whereas because the hydrogen's electrons are so close to the oxygen, the hydrogens will actually have more of a positive charge to them over here. So this creates a polar molecule, meaning it's opposites, right? When someone is really polarizing, you think that they really have those opposite views of you or other people. So that's what the word polar means. So when we say that it is being, the stimulus causes a depolarization, it means that we are going to try to take that negative environment inside the cell and bring it closer to the positive charge that's taking place outside of the cell due to all the sodium. Now let me take a step back and talk about that resting neuron one more time. We said inside the neuron we are going to have lots of potassium, and outside of the cell we are going to have lots of sodium. And we also have lots of chloride and calcium too. But let's think about the charges here. Sodium is positive, so that's why the outside is very positive. Now you might be thinking potassium, that's positive too. But the other thing that isn't fully described here in your text, because it's more of an anatomy type of topic, is that the potassium is in here because it's attracted to these really large proteins that have a negative charge. And those proteins also can't leave the cell. So that's why the inside is negative. So, like we said, when we have a stimulus, we are going to try and depolarize the neuron. So we're going to try and make the inside more positive. So the way that we will do this is by opening sodium channels. So remember we said we have lots of channels along the plasma membrane of a neuron. And so when these channels, due to this voltage change, right, we see that change taking place here, it's becoming more positive. When we hit a specific threshold, these sodium channels are going to open and allow a ton of sodium to move into the cell. We said sodium has a positive charge to it. So the inside of the cell is becoming more and more positive. Like I said, when that threshold is reached, that's when all of these channels open up. So we see a spike of positive charge, a spike of that voltage turning positive until we reach the full action potential and that information, that impulse is moving through. So that's what's being depicted down over here. This is a resting neuron. We see the inside is negative. Here's our little graph. And then as the gates are opening up due to a stimulus, we will see lots of sodium flooding in, causing it to turn positive, depolarization. And so we see depolarization moving on up to reach that action potential at the top. And then we have to reset this neuron. So we go through a process known as repolarization. We're going back to that polar environment of the cell, of it being negative inside, positive on the outside. So to do this, we are going to close these sodium channels now and instead open up some potassium channels. And the potassium channels allow for potassium to leave the cell, which we know is a positive ion, and will drop that voltage of the cell and go back to, it kind of overkills and goes below that resting membrane potential of -70. But eventually, by using our sodium-potassium pump, we bring that back to its true resting state at -70 millivolts. And that's what we're seeing here too. We're bringing that negative charge back inside of the cell and trying to get back to our resting membrane potential. Now, nerves are really interesting. They have this all or none law. It means once that impulse starts moving through and is initiated, it's going to travel through the entire length of the neuron until we reach the end of that neuron and can allow a synapse to take place. So here is my presynaptic neuron. That impulse moved through and we got to the axon terminals here and sent that information through a synapse. Remember that synapse, we're going to have a little space here known as the synaptic cleft. That is going to allow the information to travel through. Now, before the impulse traveled through a electrical type of current, right? We turned the inside of the cell positive and then went through repolarization to make it negative again until we reach the end. Now, when we reach the end here at the synaptic terminal, that voltage change is going to trigger an opening of channels that we don't see pictured here, but they are going to be calcium channels. By the calcium channels opening up here, calcium is able to move from the extracellular environment. Remember, we had more of an abundance of calcium outside of the cell. It moves through that channel and it attaches to these vesicles. Now, these vesicles contain a chemical known as a neurotransmitter, and that neurotransmitter, whatever it is going to be allowed to exocytose, to leave the axon terminal and move through the synaptic cleft here and attach to another protein channel. Now, remember when we discussed protein channels, I said sometimes we need a chemical messenger to attach to it in order for it to open up. And that's what's happening here. The neurotransmitters are serving as chemical messengers, moving across the synaptic cleft and attaching to these protein channels, allowing them to open. Now, when these channels open, what is going to move through? It's not going to be these neurotransmitters. It's going to be sodium once again. The sodium will move into the postsynaptic neuron right over here, and that causes a positive change inside of the cell, a positive charge inside of the cell. And so you guessed it, we're going to have that same thing happen where that positive charge will then start to build, reaching threshold, opening those sodium channels all along the axon, allowing the impulse to move through our second neuron. And so this synapse is more through a chemical type of mechanism since it involves neurotransmitters, whereas moving through the axon is more of a electrical mechanism. Now let's visit excitatory neurotransmitters and synaptic transmission. We said that in order to start to move toward depolarization, we need to turn the inside of the cell more positive. And this is done through excitatory postsynaptic potentials. We can do this by utilizing excitatory neurotransmitters. So let me go back one step. There we go. So imagine inside these vesicles, we have neurotransmitters such as either dopamine, or it could be acetylcholine, or serotonin, or norepinephrine. These are all excitatory neurotransmitters. Let's stick to acetylcholine since most of you are familiar with that in muscle contraction, right? So that acetylcholine moves to the protein channel, attaches to it, and causes it to open, and sodium moves in. When sodium moves in, it turns the inside of the cell more positive, And so we get that EPSP, that excitatory postsynaptic potential that is positive. Now, this can happen in two different ways. It can happen through temporal summation. That word temporal should remind you of the word time. So that means we're having a lot of EPSPs taking place close in time through one presynaptic neuron. So this is depicted over here. This is my presynaptic neuron, and it is giving you those potentials at one specific place, but back to back. Those EPSPs are building on top of one another so that we can get closer to the threshold. Whereas with a spatial summation, that means that we have multiple presynaptic neurons synapsing onto a cell body of another neuron at the same time. So we see that they are building up together. If we were to show on the graph here that these are synapsing at one time, separate EPSPs, we could see A, B, C. But if they are taking place in three different locations, hence the word spatial, they're building on top of one another, and we can reach that threshold much faster. Our goal, again, reaching that threshold, is to get that action potential to take place, to get all of those voltage-gated sodium channels to open up at one time, causing a flood of sodium to move in and the inside of the cell to become positive. Now, we can also have inhibitory postsynaptic potentials take place as well. These are going to be potentials that cause a hyperpolarization. Remember, hyper means excessive. So this is going to cause us to go in the opposite direction and make the cell even more polar, more negative. And our neurons with a more negative membrane potential resist depolarization. So I know within this graph here we are seeing some EPSPs that bring that potential up, more positive, but imagine that it was at rest and then we had an IPSP take us even more negative. Then it's going to be even harder to reach the threshold to get depolarization to take place. Some examples of inhibitory neurotransmitters would include GABA, which stands for gamma aminobutyric acid, glycine, and serotonin. Now that we kind of reviewed some of the anatomy and physiology of a neuron, let's switch gears and talk about sensory information and reflexes. We'll have some of that sensory information come in through joint proprioceptors. These are receptors that are providing our central nervous system with information about where our body is in space. So it'll be located within our joints and muscles. I may have had you do this in class before, but if you were to close your eyes, put your hands up in the air, and then asked you to use your left hand to touch the tip of your nose, chances are that you are able to do it. So if you don't remember doing this, go ahead and pause the video and try it real quick. So if you were able to do it, then you have good proprioception, right? You know where you are in space, and you've achieved kinesthesia, meaning that you have a conscious recognition of the position of your body parts. This is also called kinesthetic sense. And we also have recognition of the speed of our limb in movement as it moves from the air over to the tip of our nose. Now, the types of joint proprioceptors that we have would consist of free nerve endings, which you see pictured over here, and this is the most abundant type of a joint proprioceptor that we have. These nerve endings are sensitive to touch and pressure. So initially, it will be strongly stimulated and then will start to adapt. For instance, when you got dressed this morning, you put on your clothes, you probably had that stimulation of, yes, I'm putting on this jacket. Maybe it's a jean jacket. The fabric is a little bit more rough than my cozy sweatpants. And then you adapt and you don't have that input really cognitively or consciously within your head, right? And then we've got our golgi type receptors, and these are going to be found within our ligaments and around our joints. So I'll show you a picture of them in the next slide, and they'll be functionally similar to these free nerve endings. And then we've got our Pacinian corpuscles, and those look more like this over here, more of the complex neural receptors, where we will have them located within tissues and around joints. We definitely have Pacinian corpuscles deep within our skin. So when you go and get a Swedish massage, these nerve endings that are surrounded by these concentric layers of connective tissue will be pushed upon, and we have that sensation of deep pressure. And they'll also detect the rate of joint rotation as well. So let's dive deeper into some muscle proprioceptors. There are two primary muscle proprioceptors that will give us sensory feedback to our nervous system. We will obtain information as far as the changes in muscle length and the force development by the muscle fibers. And the first type will be our muscle spindle. So these are going to provide information about the length of our muscle and the rate of shortening or contraction. So the way that this structure here will help us with protecting our muscle is if we sense a excessive stretch within our muscle, then we will have these spindles fire rapid impulses to our nervous system, and that will result in a rapid contraction of that stretched muscle so that we don't overstretch it and cause that muscle to, let's say, tear. The other type of proprioceptor we have is the Golgi tendon organ, or sometimes referred to as GTO. And this helps to provide information about the muscle force production. So you can see this is our tendon here. That would be, let's say, of the biceps brachii. And we have an axon located around collagen fibers here. Now, when we are doing something like lifting weights, this Golgi tendon organ is sensing how much tension is in the muscle. So I always like to think of this T in tendon for tension. And if there's too much muscle tension, then the Golgi tendon organ is going to inhibit the muscle from creating any force through the reflex arch to protect you from injuring yourself. And the body does this by causing muscle relaxation before the tendon tension becomes high enough to cause damage. So let's talk about some responses to changes in muscle length using that muscle spindle. We have these intrafusal fibers within the muscle, and so these are going to run parallel to our normal muscle fibers, which we call extrafusal fibers. So essentially those are the fibers out to the side over here. And then we have our gamma motor neurons, which you see pictured in the purple surrounding this area. And they're stimulating those intrafusal fibers to contract in concert with the extrafusal fibers out here. And our muscle spindle is responsible for our stretch reflex. So, for instance, if we were to cause a stretch of the quadriceps muscle group over here, so let's just focus on the rectus femoris, then we would stimulate the muscle spindles and promote that reflex contraction. So we would be stretching the muscle if we were to hit a reflex hammer here on the patellar tendon. That then is going to send information through a sensory neuron and travel into our spinal cord. This is a slice through the spinal cord. Notice how we have a direct communication with a motor neuron here now. So this is our alpha motor neuron that then travels to that same muscle, rectus femoris, which is going to tell the muscle to contract. And we know the function or the action of the rectus femoris is to extend the leg at the knee. So that causes that knee jerk reflex where we would quickly have extension at the knee joint. And here is an example of our Golgi tendon organ at work. Remember that this is monitoring force development or tension within the muscle so that we can prevent muscle injury and excessive force generation. So we are going to stimulate a reflex that causes the muscle to relax. And this is using our inhibitory neurons that send IPSPs to the muscle alpha motor neurons. So this is also known as a inverse stretch reflex. What this is the opposite of a stretch reflex. So instead of like the muscle spindle example that we had where we are innervating and using EPSPs to get this muscle to contract, we are going to do the opposite here with our Golgi tendon. So within our, let's say, hamstring tendon over here, we are going to have our Golgi tendon organs, and they are going to send information through our sensory neuron that is going to release inhibitory neurotransmitters to cause IPSPs. That then is going to send information to this inhibitory interneuron, inter meaning between, neuron of course referring to the neuron cell, and so it's just a neuron that sits between our sensory neuron and our alpha motor neuron. So we communicate through here. Our motor neuron then is going to send that inhibitory information to our hamstrings and tell it to essentially relax. And that will help to relieve that tension that was first placed within that tendon. And what's interesting is that this is taking place when we have a vigorous contraction of a muscle, right? Where we build that tension up. So it's really helping to protect the muscle from that excessive force generation. But when we are passively stretching, the muscle can also become activated through the Golgi tendon organ and will relax the stretched muscle. Now we also have some skeletal muscle chemoreceptors that are going to be sensitive to the chemical environment that surrounds the muscle fiber itself, and specifically changes within the hydrogen ions, carbon dioxide, and potassium ions are going to give us information about the metabolic rate within the muscle. So what it does is these receptors, which are similar to free nerve endings, are going to send the information over to our central nervous system through nerve fibers that are classified as group 3, which are myelinated, and group 4, which are non-myelinated fibers. And again, it'll communicate the amount of work that the muscle is doing. And this becomes really important for the body so that it can work to keep better regulation of the cardiovascular and pulmonary responses to exercise. Next, let's talk about the somatic motor function of the body. So in order to get a muscle to contract, we have a motor neuron that leaves this ventral horn of the spinal cord. It travels through an area known as the ventral root, through the spinal nerve, and will head on over to muscle fibers in order to tell it to contract, to stimulate it, right? And so these nerves that are coming out have these synaptic terminals that end on each muscle fiber. So for each motor neuron that goes to its muscle fibers, we're going to call that a motor unit. And we can discuss these motor units as a innervation ratio. So that is really referring to the number of muscle fibers. In this case, let's say 4 for this red motor neuron. So the number of muscle fibers per motor neuron. And a low ratio in the muscle is involved in fine motor control. So you want to think about the fine movements of your hand muscles, or it could be for our extraocular muscles, the muscles that really help move your eyeball around. So the ratio for that would be 23 muscle fibers to 1 motor neuron. And a high ratio in muscles aren't going to require fine motor movements. So an example of this would be our large limb muscles, something like the the rectus femoris, in which we're going to have 1,000 muscle fibers to one motor neuron. Or you could also think of our back muscle that helps us with posture. That would be our rector spinae, would fall into this higher ratio group. Now let's take a closer look at motor units and talk about the recruitment and size principle. So when we talk about motor unit recruitment, we are discussing when we have these motor units work together in order to provide a certain amount of force. And so the size principle kind of works hand in hand with that. It means that we are going to first utilize our smallest motor units. So that's represented by this teal color here, and you could see the small motor units over here. And these are made up of what we call our slow oxidative fibers. They're the type S or type 1 muscle fibers that are really working within the aerobic respiration to contract, and we're using it at rest. Whereas if we need some more force, some more strength, we are going to start recruiting some medium or intermediate sized fibers and motor units, the motor units that control those fibers rather. So that is going to be our fast oxidative or fast fatigue resistant or type 2A fibers. And then you could see that the fibers look a little bit larger here. And lastly, if we need our absolute maximal strength, we are going to recruit our type FF, so fast and fatigable, or it's also known as our fast glycolytic fibers that are known as our type 2X fibers. So that is going to provide maximum strength in order to do whatever activity that we are trying to complete. Now, the thought behind this size principle is that with our smaller motor units, we are going to have a smaller surface area that would produce a larger EPSP, and we would be able to reach the threshold sooner, causing an action potential, which when we look at the larger motor units, we are going to see that there's more difficulty in causing a depolarization. And therefore, we figure that the smaller motor units are recruited first, and then it moves up from there. Now, as we go about our motor movement, we are going to also utilize something called a vestibular apparatus in order to maintain our equilibrium or balance. And this is going to be located within the inner ear, where we have structures like the cochlea and the semicircular canals and the vestibule. So the vestibule and the semicircular canals are going to be used mostly within this apparatus, and it's going to help maintain our head position. We are going to have receptors that are sensitive to any changes in our head position or it moving in any direction. So what that does is we have some receptors in there that are going to send nerve impulses to our central nervous system and let us know there's a change in position. Is that information linear or angular type of acceleration? And it also is going to communicate in areas where we can know to move our eyes along with our physical movement during exercise. This is especially going to be important in sports like gymnastics and diving. So let's walk through this pathway really quick. We have information coming from our eyes, those proprioceptors and cutaneous receptors from our joints, tendons, and muscles. And we also have that vestibular apparatus in our inner ear. And so this information is going to be sent to the cerebellum, which is going to have a role in coordinating our movements. And to do this, we're going to communicate with something called the vestibular nuclei. Remember, there's that word nuclei where we said it's a collection of cell bodies within the brain, in this case, the brainstem, that is then going to give us information back to our cerebellum so we can coordinate our oculomotor centers as well as our body movement by sending the impulses through the spinal cord and out the correct spinal nerve. Again, the end-all goal with this vestibular apparatus is to maintain our balance and be able to visually track the events of our movement. A majority of the brain is the cerebrum. So here is the cerebrum, and if you remember, that little brain back there that we just talked about was the cerebellum. So if we focus on the cerebrum part only, and we were to slice it, you would see that the inner workings here are all white matter. And we do have some areas of gray matter here that are representing nuclei. But around the periphery, we see all of this gray matter in here. And this is what we're talking about when we refer to the cerebral cortex. It's really where all conscious thought takes place. And it's where we're going to organize complex movement, store our learned experiences, and we have reception of sensory information. Now that is going to take place specifically within our sensory cortex, or sometimes you may have heard this referred to as the somatosensory cortex. And you could see that we almost have a map in this cortex of where those sensations are coming in from to the brain. So it's showing the moving through that white matter to reach the gray matter here in the sensory cortex. Then anterior to that, we are going to have our motor cortex. And this is also referred to as the precentral gyrus, whereas the sensory is also referred to as the postcentral gyrus. The central gyrus, the central sulcus, which is that depression right over there, sits between the motor cortex and the sensory cortex. So that motor cortex is going to allow for voluntary movement, and input is moving from the motor cortex to subcortical structures. By subcortical structures, we mean within this area over here, and also eventually will make its way down the brainstem and will communicate with the cerebellum as well that we saw back in here. And this is absolutely essential for, again, coordinating those movements, and it serves the motor cortex really serves as a final relay point upon which the subcortical inputs are focused. And then our motor commands are going to move down through the brainstem, through the spinal cord, and out to the specific area that we are trying to move. Now, in your book, there was a little informational section on what area is responsible for exercise-induced fatigue. Is it going to be due to central fatigue thought, which means we are having some type of neuronal dysfunction within our higher brain centers and or the motor neurons? Or it could be due to the dysfunction that we may have due to depletion of excitatory neurotransmitters in the motor cortex that gives us reduced motor output to the muscle. And the other theory or thought is that there's a central governor that is going to produce this exercise-induced fatigue. And that central control center is going to regulate exercise performance by reducing the motor output to the exercising muscle. Now, we think the body does this in order to protect it against catastrophic disruptions of homeostasis, essentially stopping yourself from working that muscle so hard that it causes damage. So research is suggesting that fatigue may be due to both the central and peripheral factors, but the thoughts are too that it may just depend on the environmental conditions and the type of exercise that is being performed. Now, I've mentioned the brainstem a few times already, but let's just reorientate ourselves. Here is our cerebrum, a part of our brain. We have our cerebellum back here, and look in what close relationship it is with the brainstem. Making up the brainstem, we have the medulla here, the pons, and the midbrain at the very top, the superior end. And really this brainstem is a series of tracts and nuclei that are responsible for metabolic functions, cardiorespiratory control, and some complex reflexes. Also extending throughout this area is something called the reticular formation. It's going to extend through the brainstem and also reach out and send neurons over to the cerebral cortex. And its job is to receive and integrate information from all regions of the central nervous system. And it works with these higher brain centers to control muscular activity. So one of the things that I really like to give a better idea of what this reticular formation does for us is this morning when you woke up, think about the sensations that started to come in. Maybe some light from your windows that would be moving through our eyes, right? And that's sending all information in. Maybe you had a roommate that made a fresh pot of coffee and that smell helped arouse you to be more alert, or you had an alarm clock that went off and you heard that come in. So all of those sensations come in and it activates this reticular formation and wakes up that cerebral cortex for you. Now, the second clinical application that we're going to talk about are sports-related traumatic brain injury, or TBI. This is a hot topic within research right now. And when we have a TBI, it means that we've had some type of forceful blow to the head that is going to cause damage to the brain tissue. The concussion can be mild and can typically resolve within a few days. And at times it may include loss of consciousness, but it's not always present. Your book put together a table here of some of the signs and symptoms of a concussion, so you can see some of the physical signs and symptoms that are possible, some cognitive changes, emotional changes, and sleep that may be affected as well. Now, we are going to complete a clinical application assignment on this, in which you are going to listen to a podcast by Peter Attia on concussions. And we know that this is something that people can recover from, but it can also pose numerous health risks. So some of those would include permanent brain damage or death due to brain swelling, second impact syndrome, which is when the individual experiences a second head injury before they completely recover from the initial head injury, which can prolong the treatment and for the patient to fully recover from these head injuries. We also have same season repeat concussion, which means that they have a head injury within the same season. It may be that they recovered from that first head injury, but then had a second one within the same time frame of a season. And within or between 2015 to 2019, the risk of sustaining a same year repeat concussion within the NFL was about 0.4% to 0.7% per season. And if we looked at it from a same year repeat concussion, that was an increased percentage of 5.3% to 8.3%. And lastly, we have late life consequences of repeated concussions. And this is when we have repeated concussions taking place that have been linked to neurodegenerative conditions such as Alzheimer's or Parkinson's, which we actually are talking about on this slide. So I'll leave it at that for now. But something else that we've seen is that females appear to have a higher rate of concussion than males. And the differences between sexes remain uncertain, but there are certain traits that they may have. So for individuals that have higher incidence of getting car sick or motion sickness, or if an individual has more anxiety, they tend to be a population that experiences higher rates of concussions or effects from those concussions as well. And it may relate to other physiologic and injury reporting differences. Like we said before, having repeated concussions are going to elevate the risk for degenerative brain disorders. Now, this is a little bit different than what is said on the podcast, so I want you to listen in closely for the most recent reports. And then the disease process responsible for TBI-induced dementia is called chronic traumatic encephalopathy. So they looked at these PET scans of a person who has not suffered any concussions, someone who has CTE, and someone that has Alzheimer's disease. Now, you may have noticed that the person with the chronic traumatic encephalopathy and the Alzheimer's disease brains are the most similar here, and this is associated with neuronal death, axon loss, and inflammation. Now, since we've talked about some of the motor functioning of the brain, now let's move into some motor functions of the spinal cord. We have a process called spinal tuning, in which we are able to refer to our neural networks that are intrinsic to help us operate our motor functioning better. So essentially what's going to happen is that we use our brain and those higher brain centers to send out information to tell our muscles to contract. But then specific details of the movement are going to be refined at our spinal cord level through the interaction between our spinal cord neurons and those higher brain centers. So essentially it'll play an important role in our voluntary movements. Now, something that isn't necessarily voluntary is going to be our withdrawal reflex. And this happens when we want to remove a limb from a source of pain. So imagine that you are walking along and you step onto a tack. Ouch! So this will be our left lower limb, and this will be our right lower limb. This is the way we're going to kind of discuss it. So when we step on this tack, that's painful. So we're going to send information of pain along this sensory neuron. Now notice the sensory neuron has multiple connections. We're going to send information to our brain so that we are conscious of this pain. We are going to send information along the same side, so to this same limb, by synapsing with a excitatory interneuron. Now remember, excitatory means that we're going to stimulate the muscle. We're going to get it to contract. So we need to communicate with an alpha motor neuron, which travels to our hamstring, and it tells the hamstring to contract, causing us to flex at the knee. The leg at the knee. So then we pull away from the tack, but then we can lose our balance. So the other thing that we need to do is to have the sensory neuron communicate with another excitatory interneuron that will move across the spinal cord. This is a segment of our spinal cord, right? So it's moving across the spinal cord and connecting to an alpha motor neuron on the opposite side of our body, and sends the information along the alpha motor neuron to tell the quadriceps femoris muscle to contract. When we contract that muscle, that tells us to extend the leg at the knee, which allows us to balance on one leg and continue to keep the other limb withdrawn. Now that we've talked about motor control in the brain and the spinal cord, let's put it all together so we can see how do we control for our voluntary movement. We're going to start with our initial drive to move, our plan to move. And that's going to take place in our subcortical and cortical areas. And the goal here is to have these motivational areas play a role in our consciousness of that prime drive of movement that we have. We're going to send that information to association areas, which are going to be different from the motor cortex. And this is when we have a rough draft idea of the movement pattern that we would like. From there, we're going to send information to the basal nuclei and the cerebellum. So this really also depends on what type of movement that is taking place. They do speak to one another in order to coordinate and smooth out the movements, but essentially our cerebellum is going to be more in charge of fast movements, whereas the basal nuclei will be focused on more slow or deliberate movements. From there, we are going to move into our thalamus, which is known as our relay station. Usually the thalamus is going to have more sensory information coming in, so we will take advantage of some of the information coming in from our joints, knowing where we are in space to incorporate with the movement. From there, we move into the motor cortex, which is going to be that area that we looked at a couple slides ago, where we have the body mapped out and we can execute our final motor plan. That plan will move down neurons through the spinal cord and over to our motor units to tell the muscle fibers to contract, and we will have the final movement. Now let's discuss our autonomic nervous system. So remember from the beginning of this lecture, the autonomic nervous system is involuntary. It's going to help us have a key role in maintaining homeostasis of some effector organs, such as our cardiac muscle, our smooth muscle, and those endocrine and exocrine glands. So we have two different divisions of the autonomic nervous system. And yes, we also have that enteric system, but we're going to focus here on the sympathetic and parasympathetic divisions. So our sympathetic division is going to have a couple unique features. We'll find most of the fibers here in what we call the thoracolumbar division, and you'll see that what we call the preganglionic neuron is typically short. We can see this one's a little bit longer, but when we synapse here, we are going to release acetylcholine, and then the final neurotransmitter that is released is from this postganglionic fiber to the actual effector organ. So again, that final release of the neurotransmitter is going to be norepinephrine. Now, this neurotransmitter is going to exert effect by binding to alpha or beta receptors on the effector organs. Again, this can be cardiac muscle, smooth muscle, or a gland. Once we release this neurotransmitter to those effector organs, the norepinephrine is going to be removed from the synapse or broken down. With our parasympathetic division, we'll see that we have longer preganglionic neuron fibers, and from there we are going to also release acetylcholine, and then that will synapse here at a ganglion, or sometimes it's located within the effector organ like we see over here, and then in areas where we do actually have a ganglion, we'll have that postganglionic neuron release acetylcholine, or the postganglionic neuron within the actual organ will also release acetylcholine. Acetylcholine will bind to the receptors and then will become degraded by acetylcholine esterase, which is an enzyme that breaks down acetylcholine. During exercise, the activity of our parasympathetic nervous system is going to be decreased, whereas our sympathetic nervous system is increased. And then I'll just mention also about the parasympathetic nervous system that it is going to come from cranial nerves as well as sacral nerves down here. Now let's switch gears and talk about motor unit aging and the preventative influence of exercise training. Our nervous system, as we age, of course, is going to start to decline. By age 60, our alpha motor neurons can be reduced by 50%, so that's quite a bit, and then the loss accelerates by age 75 to 80 years. Remaining motor units are going to exhibit low to moderate recruitment thresholds. So this is going to increase the muscle fiber density per motor unit. Now that's not going to give us efficient functioning of our nervous system and our muscular system, right? So we will start to see changes. We'll start to see decreases or declines in reflex times. So for example, you can kind of measure this yourself throughout the years, but if you were to have someone drop a ruler and then you want to see at what point you catch it, as time goes on, you'll see that you will move closer to the top end of that ruler. And then we are going to see declines in peak force and peak power. While our motor unit ages, inevitably, regular exercise training is going to help delay this process. So we know that exercise training enhances our brain health. It's going to improve our brain functioning and reduce the risk of cognitive impairment as we age. It's going to protect the brain from things like Alzheimer's disease and certain types of brain injury, such as stroke, especially if we are doing some great cardiorespiratory training. So how is this happening? We are able to enhance our learning and memory, and you could see with the regular exercise we're affecting areas such as the hippocampus, where long-term memory storage is one of its main functions, and we're going to stimulate formation of new neurons. Now this is still being researched within the central nervous system, but we can see more connections taking place, dendrites growing, and with resistance training, we can, of course, gain efficiency within the neurons there. We are going to have improvements in brain vascular function and blood flow. Exercise will also attenuate mechanisms driving depression. So we're going to stop that depression from taking place or reduce it. And then we will reduce peripheral factors for cognitive decline, such as inflammation, hypertension, which is high blood pressure, and insulin resistance. Now, what they found through research is that the brain and skeletal muscle is going to produce some biological factors that help stimulate neurogenesis, and improve neurological resilience to physiological stressors. So within our brain, as we exercise, we are going to release something known as BDNF, which is brain-derived neurotrophic factor. And this factor is going to be stimulated when we have acute bouts of exercise. And so when this biological factor is produced through exercise, we'll have it go and bind to our tyrosine kinase receptors on surface neurons. That forms a complex which is then going to initiate the production of cellular protective factors, leading to increases in cognition, neurogenesis, and vascular function. We also have a group of biological factors known as myokines, and these are going to be released into our bloodstream from exercise skeletal muscle. One of them is known as irisin, which is going to stimulate neuronal growth, and it's said to have this muscle to brain crosstalk that is going to stimulate metabolic pathways within the neurons of exercised brain and contribute to the neurological growth and will help prevent age-related cognitive impairment like Alzheimer's. So that is it for Chapter 7. We covered a ton of material here, so if you have any questions, feel free to reach out at any time.